is Clare Sexton, and today I am presenting on Ebola, specifically the Zaire Ebola virus. Ebola virus disease is caused by members of the genus Ebola virus. Ebola is a term specifically attributed to the Zaire Ebola virus. It is a rare and deadly disease with a mortality rate between 50 and 90%. This has led to the classification of Ebola as a BSL level 4 pathogen, and it is most commonly seen in sub-Saharan Africa, and it is also a ssRNA virus. The taxonomic information that is important to know is that Ebola virus is in the family of Filoviridae, with other members such as the Marburg virus and Quay virus, which both cause severe disease in humans and primates in the form of a viral hemorrhagic fever. It is in the genus of Ebola virus, and the various species of Ebola virus are the Bombali Ebola virus, the Bundibugyo Ebola virus, the Reston virus, which is not known to cause disease in humans and is only known to cause disease in primates and pigs, the Sudan virus, Ebola virus, the Thai forest Ebola virus, and the Zaire Ebola virus, which is the one most notable and is most commonly termed the Ebola virus. Ebola virus disease is caused by a group of Ebola viruses of the family Filoviridae, as I just mentioned, which is filament-shaped viruses. And the filoviruses are enveloped, non-segmented RNA viruses with filamentous particles. What is characteristic of this group of viruses is that there is a high mortality rate between 50 and 90%. So as we can see from this cross segment of the Ebola virus, on the outside of the virus are glycoproteins, which surround and interact as part of the mechanism for infection of cells. Within the center, we can see the nucleocapsid cylindrical shape, which in the nucleocapsid proteins, the RNA genome is stored. So the most notable strains, as I mentioned, is the Zaire or Ebola virus, and as you can see, the red dots represent this virus, and here we can see the number of cases it corresponds to based on the size of that dot. The Sudan virus, as we can see, is blue, and that is the other second most common type, and is the second type that was first identified with Ebola virus. So Ebola virus first appeared in the form of simultaneous outbreaks in 1976, the first occurring in South Sudan and the other in the Democratic Republic of the Congo in a village near the Ebola River. The Sudan Ebola virus was located in Sudan, and Zaire Ebola virus was located in the Democratic Republic of the Congo. The mortality rate of the Sudan Ebola virus was around 53%, whereas the Zaire Ebola virus was higher at 89%. So what this diagram is showing is a hypothesis of the Ebola virus transmission from animals to humans, and it is still not 100% known what the direct virus reservoir is, but right now it is currently assumed to be fruit bats. And the virus is introduced to the human population through blood, secretions, organs, feces, bodily fluids of the infected animals, such as fruit bats and other primates. Once the virus is in humans, it can be spread to other humans through direct contact with blood or bodily fluids of a person who is sick or has Ebola. It can also be spread through objects that have been contaminated with bodily fluids. And even the deceased body of an individual with Ebola can transmit the virus to other people. Specifically, healthcare workers and burial workers are at risk for this. So this is the mechanism of infection. As we can see here, step A is the Ebola virus enters. Step 2, the glycoproteins on the outside of the virus, as we saw in that previous figure, bind to the outside of that layer of the cell. Once the glycoproteins have fused with the layer of the cell, it is recognized and engulfed by the cell, and it enters the host cell genome in the host cell cytoplasm. And this is where viral replication can rapidly occur. So once the virus is inside of the cell, it kind of hijacks the host cell's proteins to create more copies of itself. And as these copies are being made rapidly, all these viruses are able to go to other cells and infect new cells and travel to other organs and areas of the body. And cause many problems there. So this looks like a complicated diagram, but it is pretty simple, just very in-depth. So as we can see, the Ebola virus first enters the body through dermal entry, through cuts, scrapes, open skin through the epidermis, or it can also enter through mucosal membranes. And it infects many cell types, but it specifically targets the macrophages and dendritic cells. The Ebola virus will then migrate from that site of infection in those macrophages and dendritic cells to the lymph nodes. And once in the lymph nodes, it spreads to the liver, spleen, adrenal glands, and travels throughout the body. The lymphocytes then undergo apoptosis, and decreased lymphocyte counts occur as a result of the adaptive immune system becoming dysregulated. The innate immune system at this time is also becoming dysregulated as the Ebola virus is taking over and just overwhelming the immune system. And excessive cytokines are released. As these pro-inflammatory cytokines keep being released, effects such as vascular leak and impairment of clotting in the circulatory system lead to severe internal and external bleeding, which will affect multiple organs and ultimately can cause multi-organ failure, shock, and lead to death. So this diagram here shows the frequency of cases in Guinea, Liberia, and Sierra Leone during the Ebola outbreak from 2019 to 2016, or 2014 to 2016, I'm sorry. And in this 2 years, we can see that 2014 had the greatest number of case incidents. And the Zaire Ebola virus was the cause of this outbreak. And approximately a little over 28,000 cases were reported, and the mortality rate was around 40%. And most of these people who were infected were in these 3 countries, but also in this outbreak specifically, a lot of foreign aid workers and healthcare workers contracted the disease. So the 2014 to 2016 outbreak in West Africa was the largest Ebola outbreak since it was first discovered in 1976. So this originated in Guinea, Sierra Leone, and Liberia, but the United States had 11 cases of Ebola, mainly from humanitarian and healthcare workers who were working in Africa in the Ebola pandemic or in the Ebola epidemic, and traveled back to America, or healthcare workers who contracted the virus after treating individuals with Ebola here. 9 out of the 11 in the United States who had Ebola virus recovered. This picture is actually a picture from the hospital where 2 nurses contracted Ebola virus after coming into direct contact with the blood of and bodily fluids of the man who traveled from West Africa to Dallas who had Ebola virus. But this hospital and Dallas in general was able to control and prevent a local outbreak because of healthcare infrastructure and proper personal protective equipment. So people most at risk for contracting Ebola, are healthcare workers and people in developing nations with poor healthcare infrastructure. Symptoms of Ebola include hemorrhagic fever, fatigue, muscle pain, headache, sore throat, vomiting, diarrhea, rash, impaired kidney function, internal and external bleeding, which is often the cause of death, low white blood cell and platelet counts, and elevated liver enzymes. To diagnose someone with Ebola virus, they need to present with Ebola virus symptoms and have a possible exposure to Ebola virus within 21 days. A PCR test can confirm this. And as far as treatment goes, the simplest treatment is supportive care and this is before the vaccine in monoclonal antibodies existed, which simply targeted rehydration through fluids and treating the nausea, the vomiting, the diarrhea. But in 2020, there were 2 vaccines that were approved, the Iverbo vaccine and the Zabdino and Maviba vaccine. And also in 2020, 2 monoclonal antibody treatments were approved for the treatment of the Zaire Ebola virus. Inmazeb, which is a combination of 3 monoclonal antibodies, and Ibanga, which is a single monoclonal antibody. Both of these bind to the glycoproteins on the virus surface and prevent replication. To prevent future outbreaks, appropriate containment measures need to be put in place, and reducing the risk of wildlife to human transmission and human to human transmission is important. Looking towards the future, upcoming treatments, it's positive to look in the past and see in only a few years how many new treatments exist, such as those monoclonal antibody treatments and the 2 vaccines. And one can expect that more treatments will start being developed, specifically for the other less common Ebola viruses. Better healthcare resources in developing countries and control against future outbreaks will help this Ebola virus not become a epidemic again and lower the mortality rate. Here are some of the sources I used in creating this presentation and the links for the images I used. Thank you so much.